So, metabolism. When we talk about metabolism in this realm of Bio 111, we're looking at mainly enzymes and then how enzymes assist with the efficiency of energy. So let's get into like what is metabolism. When we're talking about metabolism, we often think of something more holistic, like my metabolism is like how many calories I can take in and burn, and then that depends on our metabolism is like maybe weight loss or weight gain, but it really is at the cellular level. So metabolism, what do you think is the definition of it given that? So heat energy given off during chemical reactions. I mean, like maybe I see already some like, the sum of all biochemical reactions in a cell. That sounds good. Okay, the amount of energy that can be consumed by an organism. So like these two are often, like people will choose this when it's actually all of the chemical reactions in a cell. So our metabolism is what is functioning at the cellular level, and then that has a lot to do with us as an overall organism. So this leads to kind of this holistic thing here. Okay, so metabolism is the sum of all the biochemical reactions in a cell. We get our energy from the breakdown of what we consume. So lipids, fats, carbohydrates, complex carbohydrates. We'll talk a lot about, when we talk about photosynthesis and cell respiration, this is a placeholder for food in general. And I'll get into that in more detail. Our proteins, and then we'll talk about ATP, which is super duper important to our individual cells. All right, so energy. Energy is the ability or capacity to do work. So then, what's work? Cells have to do constant biological work in their, like through all the organelles. So, energy for cellular work. For example, like just moving our muscles. And when I'm talking about mechanical movement of energy use, I don't just mean like, oh, we're working our muscles out, right? So most of the organs and tissues in our body have a lining of muscle. That muscle health is not just about like working out and pumping iron. It's about exercise, even just like a brisk walk will give you exercise to your cardiovascular system muscles. So that's why exercise, 20 minutes, they say about 20 to 30 minutes of exercise per day is really critical for our health and longevity. That people who do about 20 to 30 minutes of just like I said, just like a brisk walk, or maybe some yoga. It gets just your breathing up just a little bit, gets your blood pumping. It's exercising the muscles of your cardiovascular system, which our cardiovascular system transports all the important stuff around our body. So that kind of exercise for your cardiovascular system is super important. Another just kind of context of your muscle, your mechanical energy in your bodies, the work of mechanical energy, is your digestive system. Eating healthy, eating a lot of plant materials is very important to exercise the muscles that line your digestive system. So just kind of thinking about those are two extremely large systems in our body that need the mechanical work of our muscles, and not just like these muscles. Molecular transport, moving molecules across the membrane. We talked a lot about that already when we looked at the cell membrane. Biosynthesis, making molecules and breaking molecules. And then one of the things, and we'll talk about this in a second, is the waste of energy as heat. So two types of energy. Kinetic energy is the energy of movement. And whether we're talking about like working out energy or the pumping, your blood is constantly pumping through your body, you're constantly breathing, your lungs are moving, you have the movement within your cells of, just think about like energy passing from the mitochondria to the Golgi bodies, the endoplasmic reticulum. You've got all this movement, whether you're talking about holistically or you're talking about at the cellular level. That kinetic energy is when energy is moving through your body, through your tissues, your organs, your cells. Energy, kinetic energy, is released when you break a bond. Hydrolysis. Hydrolysis is that process where you're breaking a bond. Potential energy is stored energy. We're going in the other direction. We're doing dehydration synthesis. We're storing energy in a bond. So we're constantly converting energy from one form to another in our bodies and our cells. All right, so the conversion of energy has its own set of kind of negativity around it. So let's talk a little bit about the laws of thermodynamics. The law of conservation of energy. What it says is that energy cannot be created nor destroyed, but it can be changed from one form into another. What we mean by that is if you eat something, like hopefully y'all ate something or drank something that had maybe glucose, some complex carbohydrates in it, maybe some protein, some fats this morning, and as you are sitting there and that whatever you consumed is going through your digestive system, you're having the process of hydrolysis. That food or drink is getting broken into smaller pieces. And every time what you are consuming or what you had consumed goes from something like a super macro molecule, like let's say that you had oatmeal this morning, that your oatmeal is going from oatmeal and then it's getting broken down into complex carbohydrates and proteins, and maybe you put a little bit of fat in there, maybe some like coconut milk. That every time you're going from oatmeal to these categories, and then you're making those go into from polymers to monomers, and then the monomers are going to get broken down, and eventually they go into cell respiration, and eventually they become ATP. That with every step of the way, they're getting changed from that oatmeal form into polymers, into monomers, into ATP. So your food gets converted into the currency of cells, something that's more usable to our cells, ATP. The second law of thermodynamics. Second law of thermodynamics says that when energy is changed from one form into another, the amount of usable energy, it goes way, way down. And it goes down by about 90% because as energy is converted from one form into another, energy is being lost constantly as heat. And we get these huge losses of heat. As organisms, we are what we call endothermic, that we are able to hold on to some of that heat, and we have a homeostasis of temperature in our body, and we maintain a regulated body temperature, but still a lot of that energy gets lost as heat. So we do have that benefit. The holding on to that energy as heat has been really beneficial to us as organisms because it has enabled us to go from living in hot places like Africa, where we likely originated, into other areas of the world, where we could be in cooler climates. We could take advantage of other areas of the world. But still, again, lots of that energy is lost as heat. No system is 100% efficient. No system is even close to efficient at all. For example, you might have a chemical reaction, like you put gas into your car, and let's say whatever this is, 100 units of chemical energy of gasoline, and you put it into your car, and the conversion of energy equates to, let's say, 25 units of the energy of movement, but 75% of it is just lost as heat. We know this because when you put gas in your car and you drive your car for a little bit, if you put your hand on the hood, how does it feel? Hot, right? So like after you drive your car, it's never good to think about, let's say you're going to, from home, you're like, when I get to school, I'll put the windshield wiper fluid in my car. Probably not a good idea, because you open up that engine, right? You open up the hood, and it's all really, really hot. And you know, like me, I'm clumsy. I could burn myself by just trying to open up and put in the windshield wiper fluid. So that heat, it's there, you can feel it. Most biological systems are about 10% efficient. That's it, 10%. 90% of system energy, when we're talking about most biological systems, is lost as heat. Mechanical systems can be maybe a little bit better, maybe a little bit worse. So for you all especially, if you can figure out how to capture this loss of energy as heat in some way, in some system, you'll be super rich. Because here, this is free energy that's being lost. So there was, maybe like 8 years ago, there was a kid who figured out how to capture some of a mechanical energy system to light a light bulb for like 1 second. And that was enormous news. People were like, holy cow, this is amazing. That they could convert that energy into the energy of lighting up that light bulb. And then some other engineers in the world took that system that he like created there, and they're working on trying to figure out how can we, in systems that are giving off heat, how can we convert that into the energy of things that we need in our world. So yeah, you figure that out. There's a lot of money to be had there. All right, so let's talk a little bit about this. During photosynthesis, plants convert light energy into chemical energy. This is an illustration of which one of these?
Number 3, first law.
Good, yeah, so first law of thermodynamics, we're talking about the conversion of energy. We're not talking, we didn't say anything about the loss of energy. We talked about the conversion of energy here. All right, so how do we set up reactions when you have two things that do something, or something that does something and it leads to something else? At the beginning, you have what are called the reactants, or the reactant, and then you end up with something at the end, which is the product. I like these terms, they make sense. So this is a process that forms bonds, dehydration, synthesis, we've talked about this already, or breaks bonds, hydrolysis. So that if we look at this generic equation, you have A plus B are the reactants, C plus D are the products. Chemical reactions are exergonic or endergonic. Exergonic reactions, they release energy, exit, EX. So just like we talked about exocytosis, something is leaving the cell. Here, when we're talking about an exergonic reaction, energy is leaving the reaction. It's being released from the reaction. The reactants contain more energy than the products. You can see A plus B, C plus D, and you've got this extra thing of energy here. So this is the more energetic side. An example of an exergonic reaction is cellular respiration. Glucose, let me say this again, and I like to emphasize this a lot, because for those of you who go on to take more bio classes, or if you take like biochemistry or organic chemistry, when we're talking about cellular respiration and this thing glucose. So we always use glucose plus oxygen, that when we use oxygen to break down glucose, we optimally get carbon dioxide, water, and we get energy released as ATP. Glucose, I like to say, is just a placeholder for food. That glucose, when we're talking about glucose goes into cellular respiration right at the beginning. We'll look at, when we get into cellular respiration, we'll look at what happens to proteins and fats and complex carbohydrates, that everything gets into some metabolic pathway and gets broken down into their parts, and they get funneled into some part of cellular respiration. Cellular respiration is a metabolic pathway, so it's not just like I put these two together in a pot and I stir them together and out comes this, this, and that. we're talking about a lot of reactions from here to here. And so what happens with your food depending on what this food is, it's going to get broken down. So we're talking about like a complex carbohydrate, it's going to get chopped up and then it'll go in at those chopped up pieces, various places in cell respiration, or proteins, or fats. They all get broken up outside of the mitochondria and then they start to get funneled into cellular respiration at different points. So just to know that, because sometimes I get into Bio 112 and students are like, oh, I thought we could only do glucose and cellular respiration. And I'm like, okay, really we're talking about anything goes in. Any of our energy sources get broken down and they will go in at some point. So, when we're looking at an exergonic reaction, you have your reactants, and then they react together, they make some kind of final products, and energy gets released from the reaction. Energy is exiting. Again, glucose or a food source plus oxygen gives us the release of energy, carbon dioxide, and water. So you could test this. You might, like in a biochem lab, if you take biochemistry, you might do this experiment. I remember doing it when I was in undergrad, that we would put sugar with an oxygen and some enzymes and things like that, and show that the energy initially, the heat, we're measuring the heat goes up, and then it goes down. And we get this other burst of energy at the end, so we track it. So we can do that. All right, endergonic reactions. They require an input of energy. Endo means into. Energy goes into the equation. The input of energy is at the beginning. So the reactants have more energy than the products. It's the opposite of an exergonic reaction. I mean, what did I say? Sorry. Yeah, that's what I just said. Wait, no, products. Products have more energy than the reactants. No, it should be. That doesn't seem right to me. Why does it not seem right to me? Oh, right, because energy's leaving, sorry. Because I think it's Wednesday, I'm using it. Right, energy gets long out of the reaction here. Okay, products at the end contain more energy than the reactants. You have to put in energy from somewhere else. So energy side, you have to put an input of energy plus the reactants, and then they're going to capture that energy. So photosynthesis. Photosynthesis is an example of this, that you take an input of energy from the sun, plus carbon dioxide and water. And again, we're talking about many reactions in here. That you have a metabolic pathway that eventually leads you to glucose and oxygen. The glucose can go into different metabolic pathways, like we talked about, endoplasmic reticulum, Golgi bodies, and become proteins, fats, complex carbohydrates. So we don't only make glucose from photosynthesis. If you think about photosynthesis happens for us on land, happens in plants, that plants, what it is making that photosynthesis we know is producing food. So like my tomato plant that is now like ripening and going crazy, that it's giving me my Roma tomatoes out there, and that's not glucose, right? So all of our plants, nuts and seeds and grains and fats and fruits and vegetables, all of those things, they produce food. But the plants undergo this process. So when glucose is initially made, it goes into metabolic pathways to make all of your food sources. Input of energy from the sun gets captured into the food. And you have also your carbon dioxide in your water. And you get also an offshoot product of oxygen. So again, here, energy from the sun, carbon dioxide, water, makes food, plus our oxygen that we breathe. Okay, so cells work together. One of the things that's just so amazing about our cells is that it's not like a reaction happens over here and that does something, and then a reaction over here does something, and they're all over by themselves. What happens with reactions is they come together. And so one reaction runs this way, and energy that is given off an exergonic reaction gives off energy to fuel an endergonic reaction. And then you have an endergonic reaction is going to fuel an exergonic reaction. We have these reactions that play constantly off of one another. And what we call that is we call that the coupling of reactions, that they work together. So that an exergonic reaction, energy exiting an exergonic reaction, fuels the input of energy for an endergonic reaction. This is always a cellular level stuff blows my mind, that everything in our cells is so perfectly organized and orchestrated. Remember that one of the characteristics of life is that living things are organized, and down to the cellular level. Like cellular level reactions make all the rest of us function appropriately and healthy. So every little thing we are exposed to as an organism has an impact down to the workings of our cells. The other thing is that those reactions are happening almost simultaneously. This one happens and that one happens, then another one happens, another one happens. And so they're just feeding off of each other. They're all, when we're talking about a coupled reaction, they're in the same place at the same time. So energy is stored again in bonds. And when we're talking about potential energy, we've made a bond, we've done a dehydration synthesis. When we take that larger molecule that was made or compound and we break it open, you have an endergonic reaction. So you have potential energy and then kinetic energy. So we're going to talk about, when we're looking at cellular respiration and photosynthesis, we're going to talk about specific energy carrying molecules. That these molecules are made and broken, and made and broken, and made and broken. And we reuse the parts, but the energy has to be input or exit constantly. That's why your brain, if you haven't eaten in a while, does your focus stay on what you're doing in front of you? Or is your brain like, hungry, 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 you're hungry, you're hungry, right? We start to get obsessive about being hungry because that is important for our survival. That as organisms, that when we don't have an input of energy, because another characteristic of life is living things need energy, is that our brains, as an organism that has a brain, it will take your focus away from other things that you're doing and say, go get energy. It's one of the things about the evolution of organisms is that when your survival is compromised, that whether we're talking about a unicellular organism or we're talking about something complex like us, that we have signals from our cells or organs, tissues that say, hey, 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 Your survival is compromised. Let's focus only on the thing we need to do for our survival. And so the input of energy, one of the characteristics of life, is our cells will constantly focus on that and remind us to drop everything else and focus on survival. So evolution, when organisms do not have, just by chance, they don't have characteristics or traits that allow them to survive easily, they're just constantly in the fight for survival. That they're constantly like, okay, what do I gotta do, what do I gotta do to survive? That takes away, as an organism who's not best suited, that takes them away from the process of reproduction. So if you are having a hard time surviving and your cells are saying, focus, come on, focus on surviving, does your body say, go and reproduce? No, because you're like, I gotta survive. Plus the fact that you are trying so hard to survive, do you think your brain's also like, but wait, time out. Let's bring some more organisms into this bad situation? No. So that's the process of evolution, is that when you have variety in a population, and some of those members of the population, they just don't happen to have the mutations or traits that make them best suited to living in that environment, their focus, those that without the best traits, are like, focus on survival. If I'm an organism and I'm having an easy, whatever traits I have make it easy for me to survive, I just do what I need to survive, and then I've got a lot more time, don't I? So because I have good traits for being in the current environment and I'm surviving easily, then I'm like, hey, whatever I got here, these traits, give me a lot of time so I can reproduce. And so you see the characteristics that are favored in a population change over time, depending on what traits are favored right now in the environment. That's natural selection. Nature selects for those organisms who have the best traits. So this all goes down to this like idea of what's going on with the transfer of energy within our cells, the organization within our cells, really leads to a bigger picture of biology and evolution. So, energy carrying molecules. When we're looking at, we'll look at two examples, ATP and NADPH. ATP is going to be really important when we look at both cellular respiration and photosynthesis. NADPH is a energy carrying molecule that is used in photosynthesis. We'll see that cellular respiration has FADH2 and NADH, which are different. Okay, so when we're making ATP and NADPH, the energy is held in a bond. So when we look at that ADP, ADP stands for adenosine. I don't know why I capitalized the D. Adenosine is the A. D stands for di, which is 2 phosphates. What's going to happen is then you're going to have also another phosphate. In between the two phosphates for the adenosine diphosphate, in between the last phosphate and the other phosphate that's hanging out there, you're going to need an input of energy and 2 electrons to make a covalent bond. The input of energy is going to be held in that covalent bond so that what you get is we get adenosine triphosphate, the T, the T stands for 3 or tri, because now we have 1, 2, 3. 3 phosphates. And we've made a covalent bond that's going to hold potential energy here. So we call this process the making of an energy carrying molecule, because you are carrying energy right here. In that bond. So that's ATP. When we're looking at NADPH, we have nicotinamide, adenine dinucleotide phosphate. So we take that, plus a hydrogen, plus an input of energy, plus a sharing of a pair of electrons. And again, you make NADPH, which is going to hold on to energy here, just like we had with ATP. What happens with these energy-carrying molecules is the parts, the adenosine, diphosphate plus the phosphate, the parts constantly, this will get made and then broken. So it'll get made, it'll hold on to energy, and then in another reaction later down the line in these metabolic pathways, then ATP will go this way, be an endergonic reaction. This will get chopped apart, that energy will be released and it will go to coupling with something else. So the thing about these reactions is they go back and forth within a metabolic pathway. They're not just used once or made once, that they'll get made here and then further down the line then they'll get broken. And then the parts will go back over here to get made up again, so that this ADP and this P constantly are recycled within the cell. And their job is just to get made and broken and be a transfer agent of energy. So one of the things about our cells being organized is that you have, and in this grand scheme of metabolism, is we save so much energy by reusing parts. that this gets used and then broken down. Made up, broken down, made up, broken down. So we don't have to keep constantly making ADPs and phosphates. We just use them over here, they capture energy, and then over here, they give energy, and they capture and give. Recycling of what we call our nutrients in our body is critical to lowering the amount of energy that we need as organisms and makes our metabolism very efficient. The ability of this in one place on a metabolic pathway to be made and then to be eventually broken down, it takes a lot of organization. So that, let's say that in a metabolic pathway, that this is step one, that you have to make an ATP up, and then this ATP is all the way, has to go all the way over here to step 22. You need a lot of organization to make sure that what's made here in step one goes down there to step 22. And as we talk more, it'll make more sense. But this is what we mean by energy carrying molecules. The parts get made and they get broken. They're made and broken. They carry energy and they release energy. All right, so moving forward a little bit to ATP, fuels a lot of reactions, and we'll talk about why in a bit. That's here. ATP breakdown. Got it. That here. Got the bottom of the page. Fuel. All right. So, what kind of reaction is the ATP breakdown?
Exergonic.
Good. Okay. So, when you break something down, energy exits the reaction. It's exergonic. Exergonic. Okay, so we've seen this. And like I said, that when we make ATP, it might be used in the breakdown. Energy exits and it fuels the gluing of your amino acids to make a peptide bond and form a protein. So energy gets given off here to fuel this being made. All right, when a high energy bond of ATP is broken, what primarily happens to the energy released? Is it lost as light energy? Does it function as a second messenger? Does it polarize the cell? It converts inorganic phosphates into energy-carrying molecules. That seems a little distracting. It drives endergonic reactions in a cell. Okay. Awesome. Big grand scheme of things. Through photosynthesis, what is produced, organisms like this cute little squirrel is going to eat nuts from the tree. So they're going to eat the products of photosynthesis. They're going to use the oxygen given off, also by the tree, to make the process of eating the nuts efficient. So they break that down. When we breathe out carbon dioxide, photosynthesis, what we start with in photosynthesis is carbon dioxide, water, and sunlight energy. So our waste product, carbon dioxide, the carbons, this is kind of trippy, the carbons in our carbon dioxide that we give off become our food. They become the carbons that make up our food. Yeah. The 6 carbons in glucose, glucose is C6H12O6. The 6 carbons in glucose come from our exhalation. So, what fuels what? Does photosynthesis fuel cellular respiration, or cellular respiration fuel photosynthesis? Well, we have to get that constant input of energy from the sun. So So the energy fuels the process, so we can breathe out all we want, but if there's not an input of energy from the sun, everything kind of shuts down. Trippy stuff. All right, so reactions in cells, as I keep saying, have to be orderly, have to be organized. These reactions, when we're talking about like cellular respiration, photosynthesis, other metabolic pathways in our cells, they have more than one reaction. The big ideas of photosynthesis and cellular respiration, as I said, there's a lot of little other reactions in the middle. So we call these the intermediate steps. There's a lot of intermediate steps in a metabolic pathway. If one of the steps doesn't go right, the rest of it shuts down. So everything has to be organized and orderly in our cells. Oh, here's a word, catalyzed. Enzymes will catalyze each of those intermediate steps. Catalyze means facilitate, organize, make efficient. I like to think of enzymes as, excuse me, like a party planner. That, let's say that you're planning a party for someone, your closest friend, your favorite family member. And if you decide, I can't afford a party planner, right? Then you have to go out and do every single little step yourself. You gotta get all the decorations, you gotta invite all the people, make the invitations, send out the invitations. You've got to make all the food. You've got to make sure you get all the pieces of the food to make the food. There's so many steps in that. Enzymes are kind of like a party planner, where you're like, hey, you tell them, here's what I want. And then you show up to the party and you're like, look at what has been created, this wonderful thing, right? And it's all organized for you. So what an enzyme does is when you have a reaction, you have A and B, and they're going to make C plus D, what the enzyme is doing is kind of in the middle. The enzyme is like sitting there at their intermediate step, and if I'm enzyme 1, and I go with A plus B to make C plus D, as the enzyme, my job is to constantly within the cell be going, A, come here, B, come here, come here, come here. Making sure the coupled reaction, who's going to give off energy, that ATP is going to get broken down, that ATP is there, that I've got ATP, I've got A and B. I'm gonna make sure that ATP gets broken down into ADP plus P to release energy, and I'm going to take that release of energy, I'm gonna put A and B together to make my products. And so the enzyme in that reaction is making sure that the ATP is there, A and B are there, and that we can make sure that, okay, the enzyme says, okay, ATP breaks them down, they take the energy, they glue together A and B, and they push them on. And then they're just gonna sit there and gonna be like, okay, we need another one, ATP, A plus B, go. And so as an enzyme, at each one of those intermediate steps, I've got enzyme 1 goes with A plus B, and then when C plus D are made, I've got enzyme 2, totally different enzyme, totally different party planner. They're going to say C plus D, okay, I need another ATP, or maybe I need an NADPH, and whatever is going to be broken down, they make sure that's there to take the energy and put these together and push them on. Next reaction, enzyme 3. So each reaction has a different enzyme. Each reaction has their own facilitator to catalyze, to make sure everything goes smoothly. That the coupled reactions have the parts they need, that they can facilitate the breakdown of one to fuel the making of another. By being organized, by these enzymes being there at every step, it speeds up the the reactions in the whole metabolic pathway. If the enzymes weren't there and these metabolic pathways existed, what would happen in a cell is, because everything is in constant motion, and it doesn't mean that it's in fast motion, but within a cell, you might have all those components. If enzymes were not there, then reaction 1 is just waiting for an A plus B to bump in together while an exergonic reaction breaks down. Eventually it'll happen, but it'll be really, really slow. It'll be much slower. As opposed to an enzyme being like, you come here, let me break you down, let me make you into something new, and pushes them on. So by being there, they're going to speed up chemical reactions by being that facilitator to catalyze the party. Why is photosynthesis considered an endergonic reaction? Activation energy is not required. Did we talk about that yet? Photosynthesis does not comply with the physical laws of the universe. Sugar has less energy than the sun. Protein catalysts are not needed. Low energy reactants are converted into high energy products. Okay, that's good, right? Now, when you have an enzyme who is catalyzing a reaction, your reactants, A plus B, get a different name. And now they're called substrates. So a substrate is a reactant when you have an enzyme catalyzing the reaction. So we just changed the name biologically. That infers to us we have some kind of enzyme. When you have a substrate, you have some kind of enzyme who is there to make the reaction go smoothly, to catalyze the reaction. They bind together. The enzyme, not only is it there to just make everybody's in place, but A plus B, they're like, come right here and bind to me. I got you. So they can make sure that A plus B, when they bind to the enzyme, they can't go anywhere else. They're bound, and now they can take the energy from the exergonic reaction and make sure that these two, when they're pushed out, they make the right product. So everything binds together. Kind of saw a little bit of this when we were talking about the membrane, that when you see this happening, the substrate here, some of our enzymes are in the phospholipid bilayer, that when you have a transport protein, like a carrier protein, remember that we said that a carrier protein grabs onto the molecule to which it transports? Talking about an enzyme. The enzyme grabs onto this, and when it grabs onto that, it can facilitate or catalyze a reaction to push out the new thing. Same thing. That we're talking about bigger reactions, we're talking about receptor proteins, same thing. Big thing from the outside stimulates some kind of big event to happen, like cell division, for example. Even if we're going in the other direction. So here, you have ATP, you're pushing the substrate that's going to bind to the enzyme. You need this because whatever this is being pushed into an area of high concentration. The ATP, where you get the energy from, you're chopping off one of the phosphates to make adenosine diphosphate plus a phosphate. So I always love in biology that like we keep building and some of the stuff is there weeks ago and you're like, don't notice it until you need to notice it. All right, so there's this whole thing about activation energy, that we need an input of energy constantly to energize basically the enzymes. That they're not just gonna like do their job and nobody's taking care of them. They need that energy for survival too. So regardless, energy is needed as an input, but this activation energy is different than the energy that's input like the sun. I'm gonna stop that right there and just let you know that there is a necessary activation energy, but we're not gonna get too deep into that. When you take biochemistry, you'll get into that further. So, when an enzyme binds its substrates to it, you make an enzyme substrate complex. By doing this binding, it lowers the amount of energy that is needed in the reaction. So every one of these little catalysts, right? Enzyme 1, enzyme 2, enzyme 3, by calling everything over, making it bind so they don't go anywhere, they don't go back and float out in the cell, by being that party planner, by organizing everything, pulling it close, binding it, grabbing the energy of an exergonic reaction, and pushing out the product, they lower the amount of energy. Right? If you have to plan the whole party, you need a lot of energy to accomplish everything. You pay a party planner, you're like, cool, okay, I paid the money, now all I gotta do is show up and I can enjoy. You run the party, during the party you're running around like a crazy person, right? You're like, I gotta get the food out, oh my gosh, where are the plates? Nobody has napkins, I forgot the drinks in the car, and you're running around. So having that catalyzing agent who pulls everything together, binds to it, and pushes it out, it lowers the amount of energy that the cell needs. So it looks kind of like this, that when we're talking about an enzyme and its substrates, each step is like two pieces of a puzzle. Where the substrate binds to the enzyme is called the active site. Where the substrate is actively binding to the enzyme. Our substrate, where substrates fit, they're kind of boxy, fits into this boxy binding site. They fit together. There will be so many other substrates in a cell. So other substrates can be floating around. And even though you've got other substrates, this enzyme, let's say this is enzyme 1, and these are substrates 1, like maybe 1A and 1B. But all these other shapes clearly don't fit with enzyme 1, but they're there. At any given time, there can be thousands of reactions happening simultaneously within a cell. So really important that enzyme 1 is like, I need you two here now. That's what we call the active site. They fit perfectly together. Just like if you're doing a big puzzle, every piece fitting in is like a new part of an intermediate step of a metabolic pathway until you get the final product, the entire picture together. But one piece only fits with another piece or the pieces around it. So all those little steps of putting those pieces together in a puzzle lead you to a product, one final product. Each enzyme has specificity for one substrate. Okay, so the induced fit model. What this means is that the enzyme, when it grabs onto its substrates and then pushes them out, the enzyme will change its shape a little bit. So here in our illustration, our enzyme's core or channel is closed. It binds its particular substrate. It's like, hey, you come here, fit in there. And the enzyme goes from being closed, like this, changes its shape to being open, right? You have a change from here to here, and then it goes back to its original shape. So just like when I was saying that if I'm a carrier protein, I bind onto the thing, the substrate, I push it through, it changed my shape, and I go back. And every time I catalyze a reaction, I change my shape, and then I go back to my original shape. That's what we mean by this induced fit model. It'll induce fitting together, change the shape, and it goes back to its original shape. Like here, our substrates fit perfectly in here. There is a change in the shape. The enzymes goes back to its original shape, so it's ready to bind the next substrates, but the product comes out different than the substrates. Then they go on to enzyme 2, and enzyme 2 is shaped like that. We only need a small amount of enzyme for a reaction because we keep using the same enzyme over and over again. This also saves us a lot of energy. Just like ADP plus P, we keep using those parts over again. The enzyme, boop, just keeps doing its thing. They're all like little factory workers where they can just do one job and they get used over and over and over again. Because we can use the same parts over again, whether we're talking about energy carrying molecules or enzymes, it lowers the amount of energy that we need as a cell. It lowers the activation energy that we need for everybody to run. Crazy. I just always think it's so crazy that this is all evolved from a series of mutations or mistakes. All right, so reflection on lab 7. Let's talk about what should have happened too. There are a lot of factors that can affect the speed of enzymes. So let's start with substrate concentration. This is where I get like, what? Enzymatic reactions can be 600,000 times in a second. A second. Right? Like, how can these enzymes be doing something 600,000 times in 1 second? Does that not blow your mind? And if we have a series of steps, a metabolic pathway, and everybody in that metabolic pathway is operating similarly to hundreds of thousands of times per second, how are we not just like vibrating? How are we able to sit still? I don't get it. There's that much activity. And this is just like one reaction. Remember, pathways. We have a lot of pathways in our cells. Our cells are amazing. In order to work at its maximum efficiency of this, you need 600,000 substrates every single second. So if you only have 10 substrates available every second, does the enzyme work at its maximum capacity? No, right? You have to have enough substrate in order for the reaction to work as maximum efficiency. Okay, so let's say that you have 1 million substrates. Can the reaction work faster than its maximum capacity? No. You can have more substrate, but the enzyme's like, I can only do so much. Right? It'd be like saying to you all, hey, today we're going to have a test where we're going to go outside. It's a 100-point test. You all have to run a mile in 4 minutes. Anybody run a mile in 4 minutes? I think is it humanly impossible?
It's possible.
It's possible.
It won't.
by very few humans on the planet, right? What's the fastest minute mile?
Jeez.
The world record is like 4 minutes. I could check right now.
Yeah, so even if I said 4 minutes, any of you athletes even run? What you got, Lupita?
The fastest one was 3:43.
Oh.
Well, there's a lot of high schoolers that run like 4:30s, 4:40s.
Okay, so even they can't do my 4-minute mile, right? So good, I was at least at an impossible range. So if I said to you all, you have to run 4-minute mile or 100 points, you get 0. You either get 100 or get 0. You're like, what? No, I can't run that fast, right? So same thing with enzymes. Enzymes, they could have a ton of substrate, more substrate available than they need. They can't work any faster than they can work. You can only run as fast as you can run, right? Okay, so same thing with enzymes. Let's say that your enzyme broke down. We'll talk about why your enzyme might break down. If you don't have the enzyme, it's going to really slow down your reaction. Because what's happening then is you're just waiting for the random reactants. If you don't have an enzyme, we go back to calling them reactants. You wait for the reactants and a coupled reaction to all bump into each other at the right place to go to enzyme reaction 2. It may happen, but it's going to slow everything way down. All right, let's get into, okay, so do you think temperature affects enzymes? What do you think?
Yes.
Yes.
Yeah, so too high or too low of temperature can affect enzymes. So let's think about our pineapple lab. Enzymes are proteins. Proteins are like the most important compounds in our body. Most of them. Proteins do a lot of the like heavy biological work in our bodies, in our cells. But proteins are sensitive, as are most compounds are sensitive. So other factors can have an effect on the structure of a protein. If something affects the structure of a protein, we call that denatured. That when the structure is affected, it can denature a protein. If their shape is changed, so like me here, if I'm this enzyme and my active site gets changed to this and I bind something around, will it affect the ability of me to bind to my substrate, right? So if something happens and I change my shape to this, even if the little ball substrate is bouncing here, I can't grab onto it right. So my reaction shuts down. Just like if you have an actual key to a car, let's say your boop boop thing doesn't work anymore, and you have to use the actual key, but you're like, oh shoot, half of that key, if this part breaks off halfway, and I've only got this much of my key left, can I open the car? No, right? I changed the shape of my key, I changed the function of my key. So like most things in the world, you change the shape, you change its function. So same thing here. Cell respiration and photosynthesis are long metabolic pathways, so at any point in time, if you change the shape of an enzyme, it shuts the rest of the pathway down. So a lot can go wrong. So temperature, we looked at, so let's go back to the pineapple lab. With the pineapple has a protein called bromelain. It's an enzyme that will actively break down the protein in gelatin. So you filled all your tubes up with 10 milliliters of gelatin, Jello, clear Jello. And you put pineapple in most of them. You had your control, and your control with no enzymes, pineapple, So when we are talking about the pineapple lab, when we put pineapple in, we are symbolizing enzymes that are active in that pineapple. So when you have the plain gelatin, no pineapple, there was nothing to break down the gelatin. So that control tube of the gelatin only, without having this, should have gelled immediately. That you put it in the ice and it gels up. You had frozen pineapple and heated pineapple. The heating and freezing, ideally, we should have seen that when we added frozen pineapple or we added the heated pineapple, they should have been frozen and heated to points where it denatured the enzymes. And ideally, we should have seen that those two tubes went solid relatively quickly. Now, it all depends on who heated the pineapple and froze the pineapple and what temperatures the heat and the freezing were. Because if your heated tube and your frozen tube did not go solid, that means that it wasn't a low enough temperature to freeze or a high enough temperature to heat and denature all of the enzymes in the pineapple.
I was under the understanding that when you freeze enzymes, rather than actually denaturing them, it just slows them down so much that they can't function properly. Does it actually denature them?
It denatures them, depending on the temperature.
Okay.
So there's, there's like 32 degrees freezing, and then there's like 0 degrees freezing, deep freeze. So at 32 degrees, maybe like 10% of the enzymes are still moving around in there. They're active still. But when you do a deep freeze, maybe all of them get denatured. So the temperature of which you're freezing has to be low enough to denature everything for the gelatin to go solid. Just like heating. You could heat something up, but maybe not to, let's say that the enzymes need 300 degree heat to be totally denatured. And they're only heated to 180 degrees. Some of them are still active. Make sense? Yeah, so it depends on the degree to which you're going. Low-grade fevers are great. A low-grade fever, about a 101 fever, what it does is the heat, that little bit of heat makes molecules move faster. So when you have a low-grade fever, it's making your immune system cells move around your body faster. Everything's moving faster with a low-grade fever. But we get to a limit. We get to about 108, 109, and everything gets denatured. If you get a high-grade fever, it might take you, let's say that you have, you get COVID and you have 104 fever. Your recovery, so you, 104 fever, you're going to denature a bunch of enzymes in your body. Let's say somebody else in your house, they only get a reaction to COVID, they only have a low-grade fever. Your recovery times, let's say your mom with a low-grade fever, that in 2 weeks she's much better. She's like, oh, I'm feeling normal, I got my energy back. You with the 104 fever, because you've denatured so many enzymes that your body is working to replace, 5 weeks later you're like, oh, I'm finally feeling like I have my energy back. Because your body is spending all that time recreating those enzymes. So fevers, they can really affect us depending on how high they go. Low-grade fever, you want to let it ride. You got 101 fever, and it's pretty stable. It's like sitting about 101. It means that means yay, immune system is doing a great job. Brain doing a good job monitoring the immune system. Let it ride. Do not take, I don't even want to say the word Tylenol or acetaminophen, but don't take a fever reducer if you have a low-grade fever. If your fever is just sitting at about 101 or 101.5, whatever it is, if it's just sitting there. It's not going up, 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 up. But you take your temperature, you got a 101, and then an hour, it's 102, in an hour, it's 102.5, and you see it keep ticking up, you want to take a fever reducer. Because there's some connection in your brain that's looking at what's going on in your body and the fight with whatever invaders in your body that's saying, this is not good, it's not going well. And it's trying to boost and boost and boost your immune system, but your brain isn't always thinking like, wait, stop. You're gonna start denaturing other things by keep boosting the temperature up. Sometimes we have a malfunction because the invader is taking over. So that's when you want to take a fever reducer. Take a fever reducer and your temperature keeps ticking up, you go to see a medical professional. So those are good things. I've had friends who, when they have little kids, like babies, and their baby has a fever, crying. My baby has a fever, what do I do? They're gonna die. I'm like, it's okay. So I gotta talk them through this whole thing. And they're like, I knew I needed to talk to you. And I had one friend, kid's fever kept going up and his breathing was not right. I was like, go to the ER now. And everybody in her family is like, it's fine. She goes to the ER, they're like, yeah, a couple hours, your kid would have been dead. And she's like, oh, glad I called you. So just like those are good things to remember in life. So when enzymes get denatured, your enzymatic activity goes down. Whether we're talking about freezing or heating. So in your results of the pineapple lab, if a tube goes solid, the enzyme is denatured. So like if you're ready to hand it in, you might want to look over what you wrote in your discussion, because I want to see that in your results, you're talking about enzymes that where you had pineapple, right? Remember the control tube, just the gelatin, it was just a control. It was to make sure that the gelatin would actually gel. So no pineapple means no enzymes. Where you had pineapple, you're looking at your results. Were my results solid? Did the tube go solid? That means those enzymes were denatured. So in your discussion, I want you to say, my enzymes, my pineapple, was exposed to heating, freezing, low pH, high pH, whatever they were. And the tube went solid. Therefore, the enzymes were denatured by the freezing, the heating, the blah, blah, blah. Otherwise, my pineapple that was exposed to, let's say freezing, maybe it wasn't too low of a freezing, my frozen pineapple, the tube remained liquid or semi-liquid, therefore some enzymes were still active. So make sure you are spelling all that out, that you have the variable, the pineapples, was it solid? Then the enzymes were denatured. Was it liquid or semi-liquid? Then the enzymes were active. I want to make sure I see your thought patterns, that you know that the pineapple with the variable, solid or liquid, denatured or active. I want to see that through all of your discussion.